Hello, hi everyone, I'm Sandhya, your botany lecturer. In this video, we are going to study about the transverse section of the dorsiventral leaf and monocotless leaf and secondary growth. First, we will start with transverse section of dorsiventral leaf, or you can say dicotyledonous leaf. That means in dicots means the plants which bears the seeds having two cotyledons. In dicots, the leaves are called as dorsiventral leaf. What do you mean by dorsiventral leaf? Dorsiventral leaf is a leaf where the leaf can be differentiated into the ventral surface and the dorsal surface. Two different surfaces we can differentiate. So here now we are going to study about the transverse section of the dorsiventral leaf or dicotyledonous leaf. So here the transverse section of dorsiventral leaf can be divided into three layers: epidermis, mesophyll, and vascular tissues. Okay, so and this epidermis, mesophyll, and vascular system. Then epidermis. This epidermis it is having dorsal surface and ventral surface. No, so it is also this epidermis is divided into the upper epidermis, adaxial surface, or lower epidermis, abaxial surface. So and this epidermis is covered by a thin cuticle. So we are going to draw now. Okay. This is the cuticle. This is the epidermis. This is the upper epidermis. Okay. This is cuticle, upper epidermis, or it is also called as adaxial surface. Okay, then it is also having the lower epidermis. It is also having the lower epidermis. Cuticle is there, then epidermal cells. Single row of parenchyma cells without intercellular spaces are arranged. Single row of epidermal cells are present. So this is the upper epidermis, this is the lower epidermis. Which is also called as abaxial surface. Okay, so here on the upper epidermis, the stomata are less compared to the lower epidermis. In the lower epidermis, the stomata are more in number when compared to the upper epidermis. This is nothing but the stomata. Stomata are the openings which are present in the epidermis, which are useful for the gaseous exchange and for transpiration. Okay, so more number of stomata are present in lower epidermis compared to upper epidermis. Then coming on to the tissue which is present in between the upper epidermis and the lower epidermis. That particular tissue which is present in between the upper epidermis and lower epidermis is called as, this tissue is called as mesophyll tissue. Mesophyll tissue. Why it is called as mesophyll tissue? Why? Because the term itself says meso means middle, fill means it is having chlorophyll, means this particular tissue has the cells which contain the chloroplast. More number of chloroplasts are present. So this mesophyll tissue is again divided into two types: palisade parenchyma and spongy parenchyma. Palisade parenchyma and spongy parenchyma. And both palisade parenchyma and spongy parenchyma are going to have the chloroplast, but the shape and the arrangement is different. The palisade parenchyma are elongated cells and they are arranged vertically and parallel to each other. Inside this, the chloroplasts are present. This is the palisade parenchyma. These are the palisade parenchyma, which are vertically placed and elongated cells, and they are present parallel to each other. Inside this, the chloroplasts are present. These are the palisade parenchyma. Where the chloroplasts are present. They are elongated cells, vertically placed, and they are parallel to each other. They bear the chloroplast. cytoplasm, chloroplasts are present. Okay, and the chloroplasts bear the chlorophyll, which is useful for the transpiration. The chloroplasts bear the chlorophyll, which is useful for the photosynthesis. Chlorophyll is nothing but is a photosynthetic pigment which is going to trap the sunlight and is helpful for the preparation of food materials. That process is called as photosynthesis. So chlorophyll is essential pigment, photosynthetic pigment for photosynthesis. Stomata is for transpiration. So this is about the palisade parenchyma, which is present towards the upper surface. Then below the palisade parenchyma is the spongy parenchyma, which will extend from palisade parenchyma to the lower epidermis, like this. They are loosely arranged, having large intercellular spaces. They are just irregularly arranged. Okay, they are just irregularly arranged with large intercellular spaces, like this. It's a spongy parenchyma. Because of this tissue only, the leaf is somewhat flexible, and it can move. It can be flexible. It can be moving when the wind blows. It is easy to, the leaf will be easily moving. Okay, these are the spongy cells in the spongy parenchyma. Large intercellular spaces will be present. Okay, this spongy parenchyma also bear the chloroplast, which bear the chlorophyll, which is useful for the photosynthesis. Photosynthesis is a process which is useful for the synthesis of food materials. This chlorophyll is a photosynthetic pigment which is going to trap the sunlight. So what all these are nothing but the chloroplasts, which are moving in the cytoplasm of the spongy cells or spongy parenchyma. Okay, like this. Okay, this is about the spongy parenchyma. Mesophyll tissue. These are the elongated cells vertically placed parallel to each other are palisade parenchyma. This whole thing is the spongy parenchyma. Then the vascular system. Now coming on to the vascular system. Okay, the vascular system includes the vascular bundles. Vascular bundles include the vascular tissues. Vascular tissues are what? The xylem and the phloem. So here also in the midrib and the veins, the vascular bundle is going to be present. The veins are going to vary in thickness to the in the reticulate venation of the dicot leaves. Okay, and these coming on to the vascular bundles. Okay, this is the xylem. Okay, xylem and the phloem cells surrounded by the xylem and the phloem. They are the vascular tissues. These are surrounded by the bundle sheath cells, like this. These are the bundle sheath cells. These are the bundle sheath cells. Okay, you can draw with green also. Else we'll draw with green, it will be easy. Okay. these are the bundle sheath cells. Okay, xylem and the phloem. Then they are surrounded by the, with any color you can draw. Okay, bundle sheath cells. These are the bundle sheath cells. So this is the xylem, phloem, bundle sheath cells. Okay, so this is about the dorsiventral leaf or dicotyledonous leaf. Now moving on to the monocotyledonous leaf, or you can say isobilateral leaf. Isobilateral leaf or monocotyledonous leaf. So what does it say? The monocots, the plants which bear the seeds having only one cotyledon in the seed, are called as monocots or monocotyledonous plants. They are going to have the leaf which is called as isobilateral leaf. Means it is not differentiated into dorsal surface and ventral surface. Both the surfaces are same and they are similar in color. Whereas in dicot, they are different in color and they are differentiated into different surfaces, dorsal surface and ventral surface. Here, iso means same, bilateral means only on one plane they can be divided into two halves. Isobilateral leaf, and they are not differentiated into dorsal surface and ventral surface. Okay, and this particular leaf is going to show the parallel venation. So coming on to, so it is also going to have the upper epidermis and the lower epidermis covered by thick cuticle on both, not thick cuticle, thin cuticle. You can say thin cuticle, okay, like this, cuticle. Okay, and both the surfaces are going to have both the upper and lower surfaces are going to have the stomata equal in number. If two are present in upper epidermis, the two will be present in lower epidermis. If one stomata is present in upper epidermis, the one stomata is present in lower epidermis also. This is the stomata. This is the stomata. Okay, here they are not differentiated into the guard cells are not bean shaped, they are dumbbell shaped. Here the guard cells are dumbbell shaped. Remember that thing. Okay, they are dumbbell shaped. Dumbbell shaped. Okay, the stomata. Stomata. This is the lower epidermis. This is the cuticle. This is the upper epidermis. Cuticle. Okay. Then here if we see here, monocotyledons means grasses, wheat, rice, all will be having, all are the monocots. They are going to have isobilateral leaf. Then in grasses, what happens, the veins modify themselves into large empty colorless cells. These cells are called as bulliform cells, and they're compactly arranged without any intercellular spaces. Okay, and those bulliform cells are going to have the chloroplast, and they absorb the water and become turgid. And the leaf surface will be exposed. Okay, these bulliform cells absorb water, they become turgid, and the leaf surface will be exposed during favorable conditions and when water is available. When water is not available, during water stress, what happens is the cells will become flaccid, they lose water, and the leaf will get curled. The leaf surface will not be exposed. So this is about the bulliform cells. So here the bulliform cells are present which are compactly arranged. Then vascular tissues are present. Xylem and phloem are present. Okay, first we'll draw xylem and phloem. Here xylem and phloem is present. Xylem and phloem. There are no bundle sheath cells here. Okay, there are no bundle sheath cells. Xylem and phloem. Then these are the veins, no? Like this they will be present. Xylem and phloem are just distributed in order. Then here the bulliform cells are present, which are compactly arranged. Intercellular spaces like this. Okay, they bear the chloroplast. The chloroplast will be floating in the cytoplasm. They will be moving. Cytoplasmic streaming is going to take place. These chloroplasts will be moving. Okay. So this is xylem. This is the phloem, which are the vascular tissues. No bundle sheath cells, bulliform cells are present. Adaxial surface, abaxial surface. This is about the isobilateral leaf, monocotyledonous leaf. Now coming on to the secondary growth. Secondary growth. First, moving on to secondary growth, first of all we should know what is primary growth. Primary growth is nothing but here the primary tissues are there. The primary tissues are the tissues which are formed in the primary plant body. The initial plant body is made up of all primary tissues, which is useful for the growth of the plant body. So those are primary tissues, meristematic tissues. Okay, those are primary tissues. Then what happens as the plant grows, it grows bigger. What happens? There will be increase in the growth of the stem. There is increase in the growth of the stem. Mostly in dicotyledonous plants, we can observe this particular growth and mata. There will be increase in the width of the stem and the girth of the root. So that particular growth, having the secondary tissues, is called as secondary growth. Okay, so there will be increase in growth, increase in girth of stem or root because of the secondary growth. Okay, and this particular secondary growth can be differentiated into two tissues: vascular cambium and cork cambium. Vascular cambium and cork cambium. So vascular cambium is nothing but interfascicular cambium plus intrafascicular cambium will be forming into vascular cambium. Whereas cork cambium is present in the cortex region. Okay, so first we will study about the vascular cambium. First we'll study about the vascular cambium. Vascular cambium is nothing but a, see, this is the stem now. Okay, these are the vascular bundles, isn't it? This is the pith. This is the phloem. Okay, this is the phloem. This is the xylem. During primary growth, the primary tissues are protoxylem. Then there will protoxylem and metaxylem. Then in the secondary growth, they will be forming into the secondary xylem and secondary phloem. So from where they are formed, see, the tissues are going to divide in between the phloem and the xylem, and the thick tissue is going to form in between these vascular tissues. It's a cambium. Okay, it's a secondary tissue. So this particular cambium is called as intrafascicular cambium. It is going to form in between the xylem and the phloem. It is intrafascicular cambium. Then after some time, after few more years, as the tree gets older, this is going to get completed. A ring is going to be formed. Okay, so the cambium which is present in between two vascular bundles is called as interfascicular cambium. Okay, so together they'll form into the vascular cambium. Okay, this is the vascular cambium. This is the pith. Parenchymatous pith. So this is about the vascular cambium. Then if you see, there are two types of woods: spring wood and autumn wood. During the favorable conditions, when the water is available, the plant is going to grow, isn't it? So during that period, the xylem is going to have more number of vessels and having wider lumens. Okay, so the xylem vessels are more in number, and wider lumens are present. So such type of wood is called as spring wood. Or you can also call it as early wood. Okay, then what happens during unfavorable conditions during autumn season? What happens? The xylem vessels will be less in number and narrow lumens. The xylem, less number of xylem vessels and narrow lumens are present. Such type of wood which is formed is called as autumn wood or late wood. So this spring wood and autumn wood is going to completely forming into a ring-like structure. Okay, during one season the spring wood is going to form, during the other season the autumn wood is going to form. So in 1 year it is going to completely form as one particular ring-like structure. So that particular ring-like structure is called as growth ring. So this is going to form into the growth ring, or you can say annual ring. So by counting the number of rings, by counting the number of annual rings, we can estimate the age of the tree. So that particular study, the study of estimating the age of the tree by counting the number of annual rings, is called as dendrochronology. Okay, logy means study. Okay, so what is this? It's a branch of science Science which deals with the estimation of the age of the tree by counting the number of annual rings is called as dendrochronology. Okay, and here the spring wood is lighter in color. The spring wood is lighter in color, so low density. The autumn wood is darker in color and high density. Coming on to the heartwood and sapwood. Another one in the vascular cambium itself. It is having heartwood and sapwood. Heartwood is the innermost central region of the stem, where it is dark brown in color because of the deposition of the tannins, resins, gums, aromatic substances. It is darker in color, and it is hard. It is hard, durable. It is hard, durable, and dark brown in color due to deposition of secondary metabolites, deposition of tannins, resins, alkaloids, all such type of things, etc. It is dark brown color, hard, durable, and it is resistant also. It is resistant, and it is a dead tissue. It is having lignified cell walls, and it do not conduct the water at all. Do not conduct water at all. Such type of wood is called as heartwood, and where it is present in the innermost region of the wood or stem, or you can say the central core is nothing but the heartwood. Okay, it do not conduct the water at all. So what does it do? It will give the mechanical support. Coming on to, it is also called as duramen. Okay, coming on to the sapwood. Sapwood is also called as alburnum. It is present, it is lighter in color, the peripheral portion, the peripheral region, okay, lighter in color, peripheral region of the wood, and it is going to conduct the water. So such type of wood is called as sapwood or alburnum. From root to leaf, it is going to conduct the water from root to leaf. This is about the vascular cambium. Coming on to the cork cambium. Cork cambium is a tissue, which is secondary tissue, which is going to be present in the cortex, in the cortical region. So if we see the transverse section of stem, so this is a stele inside is stele. So in this stele, this is the this is the vascular cambium. So this cork cambium, this is the vascular cambium, which is formed by intravascular cambium and intervascular cambium. Okay, this is the stele. So this cork cambium is going to be present inside the cortex. Okay, this region is called as cortex. This is nothing but the epidermis, isn't it? Transverse section of stem we have seen. So this is the cork cambium. So this cork cambium is going to cut off the cells into outer region and inner region, inner to the cortex and outer to the cortex. So this is outer to the cortex and inner to the cortex. Okay, so it cuts off the cells. The new cells which are going to be formed. It will increase the width of the stem. This cork cambium is also called as phyllogen. Okay, so the outermost division of the cells towards the epidermis, that particular thing is called as phellum or cork. So this one is phellum or cork. Then the innermost, the cells which are going to cut inner to the stem, to the central portion, innermost region, it cuts off the cells now, that is going to form into the secondary cortex or the phylloden. secondary cortex or the phloem. So the phloem, phloem and the secondary cortex phloem is called as the periderm. So what is periderm now? So here this is going to be the phloem. This is going to be the phloem. So these three together is called as the periderm. Got my point? So periderm is the phloem, cork plus phloem, which is the cork cambium which is present centrally, plus the phloem, which is the secondary cortex. So careful. Okay, which is the secondary cortex. So periderm is the phloem, phloem and phloem. Phloem, phloem and phloem. Periderm is nothing but that one. Then due to the activity of this particular cork cambium, it builds up the pressure. It builds up the pressure onto the outermost region, peripheral region. So what happens, because of the pressure build up, the peripheral to the phloem, it is going to break off. It is going to break off and dead tissues are formed. So that is nothing but the bark anamata. Okay, bark. So bark is a non-technical term that refers to all tissues exterior to the vascular cambium. So from vascular cambium to the epidermis, what all tissues are there? That is nothing but the bark anamata. Okay, that is nothing but the bark. This bark is also divided into early bark, which is formed in the early growth, early of the season, early bark, and late bark, which is going to form at the end of the season. Late bark is going to form at the end of the season. Early bark is going to form at the early of the season. So what is a bark now? It's actually a non-technical term that refers to all the tissues exterior to this particular vascular cambium. This is the vascular cambium, no? This is the vascular cambium, no? So this vascular cambium, what all tissues are there towards to the peripheral region, towards the epidermis region, this all tissues are nothing but the bark anamata. Bark. Then what happens, the phloem is then the cork cambium is going to cut off the outermost cells anamata, outer side of the cells of the cork cells, and they form into here what happens, because of the pressure build up, here the epidermis is going to get ruptured. And these parenchymatous cells, soon they are going to rupture the epidermis. So here what happens, lens shaped cells are going to be present. These lens shaped cells are nothing but the lenticels. So these lenticels are present in the bark. They are the parenchymatous cells, loosely packed, loosely arranged parenchymatous cells, which are formed because of the rupture of the epidermis. And they are the cells which are they are the pores which are useful for the gaseous exchange, and they are present in the woody stems. Remember, they are present in the bark of the woody stems. So lenticels are the cells which are present in the bark of the woody stems. Okay, bark of the woody stems. This is about the cork cambium in the stem. What happens is secondary growth is also present in roots also. In dicots, in roots, the secondary growth is going to be there. They are going to form into protoxylem, metaxylem, protophyllum, metaphyllum, like that. And this secondary growth is also present in roots and stems of gymnosperms also. So in angiosperms and gymnosperms, they are present. In dicots, both in roots and stems, they are going to show the secondary growth. But in monocots, the secondary growth is absent. Okay, so in angiosperms, dicots are going to have the secondary growth both in root and stem, But in monocots, the secondary growth is absent. Both in root and stem, in monocots, the secondary growth is absent. In dicots, the secondary growth is observed in roots and stems. In gymnosperms also, the secondary growth is observed in root and stems. Then root and stems. Yes. Thank you.